Dear participants, welcome again to the technical meeting 2021 of GCRSC. Day 2 today. GCRSC, for those of you who are not familiar or members of GCRSC, GCRSC is the Global Council for Innovation in Rapeseed and Canola. So it's an international assembly of experts in rapeseed who are dealing with actual topics of rapeseed research, having different committees from agronomy to the use of rapeseed products, and GCRSC has its technical meetings every 4 years, and in between there are the international conferences, the rapeseed congresses. Last has been taking part in Berlin. The coming one in 2 years' time will be in Sydney, Australia, organized by Rob Wilson and his colleagues. And we all look forward to that conference. And today is, of course, a smaller meeting, the so-called technical conference. And today, on the second day, we are dealing with another topic. And I mentioned already yesterday that the global production of oil crops, total oil crops and vegetable oil, is continuously increasing. This means that plant oil is an important commodity. Leading oilseed is soybean, of course, followed by rapeseed and sunflower seed. And it's important to mention that rapeseed cultivation has stagnated recently somewhat. Correspondingly, rapeseed harvest in the European Union increased only moderately, while record harvests have been obtained for soybean and sunflower kernels. I want to mention here that average rapeseed yield in the European Union this year has been as low as 32 quintals per hectare, which is really low. Maximum yield obtained average yield has been 4 tons, 40 quintals per hectare a couple of years ago. Total harvest in the European Union was 17 million tons, which equals to about a quarter of world productions. Reductions, as mentioned, are thought to be due to limitations of nitrogen fertilization and also restrictions of chemical plant protection, along with susceptibility of rapeseed cultivars to pests and pathogens. In view of long-term perspectives, intense breeding efforts for improving nitrogen efficiency and pest resistance would be urgently needed. This challenging situation for cultivation and production of oilseed rape deserves our continued activity on improvement of future rapeseed cultivars for disease and pest resistance, as well as enhanced tolerance to environment stress. The need for scientific research and development in this area is very obvious. Therefore, we had chosen insect management as one of the 2 major topics of this conference held yesterday, and very well organized by our colleague Sam Cook. Increases of production should be complemented by efforts to improve seed quality by breeding and processing. Canola and rapeseed oil today is known and highly estimated for its outstanding food quality and value as feedstuff for biodiesel. Additional value could be added to canola meal by improving protein, not only as an animal feed, but also as a basis for valuable human consumption. For that reason, rapeseed protein was selected as the second major topic of this meeting. As an expert in the field and beyond, our colleague Veronique Barthé from Winnipeg has been kind enough to organize this session, and we look forward to this scientific session now. Thank you.
Thank you so much, Wolfgang, for your introduction. It was a very interesting meeting yesterday, and I hope the meeting today will be as good as and interesting. Again, like yesterday, people can ask questions. People from the audience can ask questions by raising your hand, and we will unmute you to ask the question. And we will do as yesterday. I will make a short summary of all the session, and we will have discussion with the member of the panel. So just a couple of notes about the meeting yesterday and the presentations. So actually, that was something that Soren said, that we need to increase the production of protein by quite a lot, by 50 to 80% in the next decade to feed the whole population. That's something Soren mentioned, but something that's been mentioned several times when I've been going to meeting and food meeting. So this is not something new. This is something that we know. But unfortunately, we're still talking about it because we haven't been able to solve the issue. If I look at Canada in the last 10 years, and if you look at the average, even if we have a 40% oil and up sometime to 50% oil in the seed, we still have more than 20% protein. And that's about 44% of the meal, that you kind of waste because it's not used, and it could be a very interesting sources. In most of the country, actually, when I was presentation, there was presentation from Australia, there was presentation from Canada, and there was presentation from Europe. We see that for the last couple of years, oil content has been increasing and protein has been decreasing. And that's something that's part of the oil seed and something that we have to fix. And actually, even in soybean, which is a crop that's been grown for protein, if you look over the last 10 years, there is a huge decrease in the protein content of the soybean. So there is breeding effort that has to be made in Canada. There's breeding effort that made to increase the protein content, and there is several options, and one of them is to decrease the fiber. And then there was talks about processing, and then talk about nutrition. And when we talk about nutrition, fiber is important, but also the taste. And there was some mention of tasted. So if I look at the speaker, and the first one was Dave Disiak, and he was talking about how canola is a very interesting crop actually to make protein, because it's a very sustainable crop. It's grown in all the continents. We have it in Australia, we have it in North America, we have it in Europe, and none of the protein is really used, and we should be able to do something because we have sustainable production in all the continents. And then he was also mentioning that, again, food-based plant is not as we say in Canada, the flavor of the day, but it's a trend that's going to be there to stay because we need it for feeding the chicken or the fish, but we also need it to feed the humans, and more people are turning to plant-based food. The market usually is driving the breeding, and he was mentioning that at one point, when you look at the evolution of the breeding in Canada, the breeding always been responsive to the market, and so they probably will have a breeding pushing a push of the market to breed from higher protein. And then he was at the end mentioning some of the process that they're doing in his company and the protein isolate that they were doing and the market that we're targeting, that was aquaculture, because in Canada, in North America, there is a lot of salmon farms, and we always talk about the need of a good protein source for the salmon and also for human. So the second talk actually was Rob, and Rob Golden was talking about the protein model that he's trying to develop. When we register grain in Canada, we know the quality of the grain where they breed, but we had a hard time predicting the quality of the grain on farm. And Rob is trying to develop a model knowing that what you have when you have the seed, and then when you go it on farm, what it will be and what will be affecting the thing. So he was talking about using some of the data actually that we share with them. And then some of the data it's also got from sharing with Corteva. And environment is a big part. And when they was looking at the producer samples, there was 70% of the unknown that was acting on the protein content, and that was well decreased when you were able to control and know all your parameters. And something actually that was quite interesting for me was the effect of the plant density. And I think there was a couple of questions also in these things where people are talking about plant density and the effect on the quality of the protein and the quantity of the protein. So that was quite interesting, and I'm very glad that it was showing things like that. Then we have Amin, and Amin remind us actually that when you're talking about protein, this is not new. And Dave mentioned that's not the flavor of the month. And well, actually, if you look at Kip Downey, that was in the 70s that he was talking about you need to reduce fiber, glucosinolate, and polyphenol. And that seems we still talk about it now in, like 40 years later. And so protein is there to stay, and we need to do something. And Amin was telling us, well, actually, it takes 10 years to develop something. And that's so even if the market is pushing for high protein, before we actually get something with very high protein, it will take about 10 years. So that's something that we have to be reconnaissance when you do genomics and genetics and breeding. And it was also something very interesting, and I know about it, and it was talking about the taste of the product, and it was very interesting actually, because it was a very small amount of that derivative, the camphorol derivative, that was producing the bad taste. And that was something that's also very interesting and very difficult to breed, because you need to be able to analyze it in the seed to be able to breed for it, or breed to, unless you know the gene that's doing it. And so that's something also interesting. Then then we had Zhongzhu actually that was talking about creating new gene pool and increasing the gene pool of the Brassica napus. And then I resumed this work in 1, 2, 3, 4, 5, 6 parts, 6 sentences. And so 6 sentences actually take 10 years. So when we talk about genetics and we talk about improving breeding, it could be resumed in a very small short sentence, but actually the time to do it is enormous. And all that work, it was about 10 years of work behind. And we have to also, as quality people that just dealing with quality and protein and all the things like that, we need to be, we need to be reconnaissance. We have to be in the background knowing that actually, if you really want to increase the protein content by 1%, it will take about 10 years maybe to increase it. And then we have Gertjan Smolder that actually was also talking about processing, and he was talking about his new process that they're developing in DSM, and the protein that they got, and the protein isolate that they got actually was very interesting. 90% protein with a very good PDCAS, and it was an excellent protein source. And what was also very interesting with this protein that they were doing was the quality, the nutritional quality of the protein, but also the functionality of that protein that could be used for multiple things, because they were putting it in cream cheese, they were putting in dairy, but it was also be able to extrude it and then make it something better. So that's also showed not only that you could have good nutritional protein from canola, but you also can have big good functionality protein and do a lot of things with those protein in canola. And then we have Richard that talked about Australia and what was done in Australia. Also, the whole things that's been doing, like looking also for genetics and improving the genetics by looking at potential. And then when you have the genetics, actually looking at the quality of the meal to be able to feed it and feed it to different animals to be able, and rumen to be able to establish good data. And at the end of the day, you have to feed it to feed grass, because Australia, they might need to have supplemented meal for the feed grass. And that was also something quite interesting. And last not the least, and that may be the story that we remember there is still things that we need to improve, because that was an interesting work and a lot of work presented by Soren. But at the end of the day, piglet look, there is a lot. We have a lot more in common with piglets and pigs than we have with a lot of things, and the taste and the nutrition quality of the protein in this project doesn't replace the milk, and milk is still number one. So that's mean that there's still some effort to be made on the breeding, because we have to remove probably some of the anti-nutritional compound. We also need to remove some of the tasting compounds, like the phenolics and the synapine and all those negative effect compounds. And we're still not there when we want to feed to some of the sensitive animals like the piglets. And that's also kind of go back to reality. We need to do more work, and we need to do more work on the breeding. We need to do more work on the processing, and we need to have a better understanding of exactly the rapeseed protein to be able to use it and replace it in something as sensitive as piglet. And that's about all the talks. And then I will open the discussion for everybody. And I will be happy to share your discussion and have a very good discussion as we had yesterday. So actually, Wolfgang already has the hands up. Go ahead, Wolfgang.
Question for Rob Gulden, who presented variance analysis of protein content and yield, and was interesting to see there was virtually no genetic variance, and main factors were very low also, mainly interactions. Is that typical for protein content and yield? I thought in other countries, genetic variation was larger in experiments reported from there.
Sure, give me the first question. Yeah, that's a really good question. And I think it depends on how large your data set is as to how important that genetic versus environment side is on the variation. We can make that genetic effect quite large if we use very few environments that are where the environments are relatively similar. But as we broaden our environments, particularly in Western Canada, but I'm sure the same would happen elsewhere. But in Western Canada, we're talking about a very large region. And as we increase the number of environments, that affect the proportion, the partitioning of variance towards environment steadily increases. It's not to say that genetics aren't important. It's not to say that that genetic genotype by environment interaction doesn't occur and isn't important either. But at the same time, we have to recognize that wide range of environments does become a really major driver of overall protein content. And the same happens with yield as well. And you would see that at other locations in other places in the world as well.
I mean, you have a question, please go ahead.
Not a question, but perhaps just a comment on the data that have been presented by Rob. I think all the data that has been presented are based on commercial hybrids. Then we know that in Canada that you should have a threshold of protein content there, and the variation between the hybrids and in the protein content was not so high. So I think this is why the low genetic variance that you do have then in your data set.
To some degree. Now, the data set that came from Corteva is principally their entire breeding lines. And so it's beyond, there's more variation within that data set than just commercial hybrids. On the farm side, absolutely. On the farm side, that's commercial hybrids only. Having said that, we see a large range in protein content among those, and those commercial hybrids. About 50% of the acreage is only about 5 of the 5 hybrids, and so again, there's that huge environmental effect. But on the data set that we did the more thorough analysis on, it contained 420 some odd genotypes, and most of those are breeding lines. Very few of those are actually commercial hybrids. So So I wouldn't say that we didn't have genetic variation, but it is within one breeding program.
Rob, since you were the one talking, actually in the chats earlier this morning, there was a couple of questions, and there was somebody was, Dr. Gupta was quite interested about the relationship that you show with plant density and protein content. And actually, I was also quite interested in it. And then, so they were thinking, they were asking how you could interpret that. And then you was also mentioning that you were measuring quality by NIR. So you probably also have all content. And he was wondering if you add heel. So if you can comment on those one, please.
Yeah, that's an interesting relationship. And to be honest, I looked at that data and found that relationship about a week and a half ago when I was able to pull all the data together. So I haven't had a lot of time to think about it. We certainly have that. We certainly have the data for oil content as well, and I've done a very quick and crude analysis. And it seems that the relationship, at least at the 4 locations we've crunched the numbers on, the relationship in protein seems to be more consistent than a relationship in oil content. But again, very few locations at this point, so I wouldn't hang my hat on that yet. What's happening? That's a really good question. To me, it could be a number of things. And if you look at that relationship that I showed on that graph, really, the numbers really move at 20 plants per meter squared or lower, which is typically not what we see in commercial production fields. But that's where we see this large range and this very quick drop. To me, it has to, obviously, I think it has a lot to do with the time, that maturation period in canola, right? And I showed that image where if we have high density stands, we have, one rachis, a couple of branches, and flowering happens. Flowering starts around the same time in no matter what density we have in terms of canola stand densities. However, the flowering period and then period maturation is extended by a fair bit in lower density stands. And so how that, my physiology is not good enough anymore, and I need to do some reading up on that. But how that extended maturation period maybe leads to better translocation of nitrogen to the seed, more protein formation. I'm not entirely sure. Maybe it has to do that as we get later into the season with that longer maturation period. We're now getting into cooler temperatures already. It's a combination of things. And we really, it's a very interesting phenomenon, and we see it in other crops as well. I mean, we have in my lab now data for wheat and data for field beans, and in wheat, the similar thing shows up. I have not had a chance to analyze the field bean data, but it's somehow related to that. But which of those factors is the most important one, whether it's cooler temperatures, maturation, et cetera, I can't say for sure yet. Hope that helps, but that's not the clearest of answers.
No, at the end of the day, that's what's happening, more research, because, and I thought it was very interesting because we the last couple of years in Manitoba, we had frost, and that affects plant density, so I thought that was interesting even for, in Manitoba. Can we look at it in Manitoba? There was another question from you actually, and if you were looking at oil content, and if you were the same parameter affecting the protein, did it affect oil? And then if you were looking at other parameter, like maybe disease, is going to affect parameter like that?
Great question.
Planning to do it in the future,
Yeah, no, those are great questions. So the oil side, we do have the oil data. We just have not had time to model that yet. So that's in the works. So once we get the data set built, particularly with the environmental variables, and we still have a lot of calculating to do to populate that model. We can recalculate some of our environmental variables. We haven't completely sorted that out yet. We also still need a bit more environmental data. We can certainly get soil data from the various locations that we don't have yet. So we can include that in the model, which will help. explain some things. But yes, oil content wise, certainly we'll use the same parameters to model oil content. And whether diseases affect that, I would expect so, yes, because if your diseases affect maturation in any way, a heavy infestation, then it will certainly affect seed quality, I would think.
And I have, you're really very popular. I have another question for you, actually.
Great.
Then after that, we switch to somebody else.
Thanks.
What will be, why high temperature during filling and high nitrogen affect protein? And are they both influenced by oil? Have you removed the oil content as a covariate for your analysis?
No, we haven't removed oil content. We're just talking seed protein content at the whatever 20+% range. So we have not removed oil content from that. Sure, we can do that. What was the second part of the question? Sorry, forgot already.
So the high temperature and the nitrogen and high nitrogen. Right.
So the high temperatures, and that's been documented. Certainly, temperatures above 28, which is interesting how that shows up in the model. So we observed in this model that on the temperature side, the most important driving factor is days above 28 degrees within the last 7 weeks before the crop matures. So that would presumably be close to flowering or maybe even just before flowering. Unfortunately, we don't have flowering data. So we can't nail that period down as well as we would like. But it's been shown that we do get blasting and flower abortion at these higher temperatures. Now, whether this is directly related to flower abortion or whether it's just the warmer temperatures, the other bit that I find interesting is going to take a little bit of snooping through the data set to better understand what's going on is this other part to the temperature side, which was that presumably warmer night temperatures also have a negative effect on protein content during that 7-week period before maturation. So those two components still need to be sorted out. I'd like to see, and we've done some really crude correlations. Unfortunately, with a data set that has 15,000 data points, you get significant correlations between almost everything. But whether they're meaningful is a different story. And so we still need to sort out whether these things are actually meaningful or not. There's a previous study done by Taryn Dixon, who also showed that it was much smaller. It was all within 100 kilometers of Winnipeg. But they also showed that these temperatures during maturation are very important, the high temperatures. Physiologically, whether that slows or speeds up the maturation process and doesn't allow the plant to translocate as much nitrogen into the seed or form protein, however it does, I'm not quite sure exactly what's happening there. And that certainly warrants further investigation or certainly inspection of the data. But yeah, there's some interesting things we found, but it's this nighttime temperature bit that I'm also curious about what the relationship there is, because that's one that's negative. Nitrogen fertility, we still have to sort out that piece completely. Over all these environments, we have a really large range of nitrogen fertilizer that was applied. What we have not added into that equation yet is because we don't have the soil data, we do not have residual nitrogen variable amounts calculated into this. So the question to me is, will residual nitrogen actually matter when we have those values added in? Or is canola just that much more responsive to mineral nitrogen additions of that season and perhaps less responsive to residual soil nitrogen? I don't know. That is something that we still need to look into and are going to look into.
Thanks. And we have a question from Etienne, actually. So please go ahead, Etienne.
Yes, about this relation with plant density. In fact, I was wondering if there could be an interaction with fertilization and nitrogen fertilization. I mean, not a single effect of nitrogen fertilization or plant density, but a mix of the two, in fact. Is to say, leading to differences of nitrogen availability per plant.
Could very well be, yes. The studies that I've based this on, we have all fertilized to exactly the same level. Every plot has the same amount of nitrogen applied. We have, however, the plant spatial arrangement is very different in plots. We have row spacings, up to 6 different row spacings, and then 8 different densities. So you're right, there could be a piece of nitrogen availability in this. Having said that, I don't see, and I didn't show the data, but that graph where I show that relationship, that actually includes all the row spacings combined together. And if you try to separate them just visually, I've not done it statistically yet, but just visually, there doesn't seem to be a strong row spacing effect there. It's mostly driven by density, which is quite interesting because the yield response, well, it's also mostly driven by density, but on the yield response with respect to row spacing, we see that any row spacing that's below 30 inches or 75 centimeters in our environment, we get the same yield plateau. But as soon as we widen them more than that, we actually start dropping yield, which is, it speaks to the plasticity of Brassica napus. It is quite a fantastic plant in terms of how well it is able to capture space. But there could be a piece of that in there. You're absolutely right. And we do need to look at that a bit more closely. And once we get more of that data collected, we'll be able to do that. Agreed. Yes, but there, unfortunately, in that experiment, we don't have a nitrogen rate component to that. But we have another study where we actually have that's being conducted in conjunction with Ag Canada that's looking at nitrogen use efficiency of 2 cultivars, but not density. So maybe once we combine all this stuff, we can piece these things together anecdotally.
Thank you.
Thank you. I have a question actually for Gretchen. It's for the processing. And one of the question was, can you process other brassica than the canola? And if the, the anti-nutrient of the other brassica, if you look at Meston, for example, Brassica gentiana, will be affecting your process and so on.
Yeah, we do not have a lot of experience with that. But in general, everything that is close to Brassica napus, if you look to the mustard varieties, the glucosinolate levels are much higher. And they can really be a lot higher. So we have never tested that in our process to see the effect of removing it. But I guess that with the removal methods that we have in place, you can come quite far. But it is not said if in the end you will be sufficiently removing the mustard taste, because that's what you will have to do, because otherwise you will have a protein that you cannot consume in large quantities.
Yep. And then I had another question. There is a couple of questions actually about processing and the
okay, good.
And what will be the economic viability of Canola Pro compared to other protein isolate?
Yeah, it is as a result of the processing, it is a higher priced protein, but it is also offering a very unique functionality and nutritional profile. To start with the nutritional profile, it is the best plant protein around, I think, together with potato protein. And that is because all the essential amino acids are present in sufficient amounts. And that's really unique among plant proteins. So you can use it in all kinds of very specific applications, sports nutrition, elderly nutrition, and so on. And towards the functionality, since it is soluble over the full pH range, it's much more close to a whey protein. Also could think about prices in that range. So it's clearly not on the price level of soy. We also do not want to compete with prices of soy or people that are happy with soy. We really look for higher prices than soy and pea.
And Wolfgang has a question also.
Yeah, again, to this presentation of Gert-Jan Smulders. You have, as far as I recall, you have not mentioned the cruciferin relation of the protein compounds or components which Armani Abadi has mentioned. Would that be important for your product?
Yes, of course, that is important. Our ratio is roughly 60% mapins and 40% cruciferins. But you can influence that by how you process it. But it is also interesting. It is an interesting breeding question, I think. And the question is then, to what extent can you influence these ratios? Because you could think of variety where you increase content, or the other one, the cruciferin content.
Thank you.
Sufficient answer for your question?
Yes, thank you.
Okay.
So actually, there was an interested question, and when I say interested question, it's because it was a money question, and then it was somebody that was asking like, will you be, since you're looking for probably high protein content canola variety and people producing high protein content, will you be paying more for those high protein content from a producer?
Yeah.
Will you give premium?
There might be, of course, there can be a premium for high protein varieties. This is also one of the last slides that I had in my presentation, to express the need for high protein varieties, because that will really change the value chain of the whole canola protein or the canola processing. And of course, if there's more protein inside the seeds, you can afford a premium to that. But I cannot already give directions how much that premium will be. So.
Actually, there is Dave Diziak. That was one of the speakers. He's not there on the panel yet. No. But there is, it was working for Dalco, that's not Corteva, and they were developing a high protein, low fiber variety that's grown into IP system in Canada. Yeah. But it's a GM. So my question as a Canadian, where we're growing 90% of the crop, which is GM, there is 10% of the crop that's not GM, but most of it is still GM. How do you see this variety being able to be used in a system like you have, or will it be used for food because it's a GM crop?
It is a matter of choice, of course, which way you want to go. For Europe, it's very clear, and we are of course based in Europe, so we had to work with non-GMO because it's simply not allowed, and that also limits our choice largely in high protein varieties, because I think in Canada, indeed in the GM range, there are some very interesting varieties, and if you do not bother to have a GM protein in the end, then it's very attractive. But you see that in the market, the trend is really moving towards non-GM. So the question is how to get non-GM varieties still with high protein. But it is for Canada a small market still, but it might change over time.
And another question also, when you're looking at, because you need to have high protein, but actually you need to have not so much high protein in the seed, but high protein in the meal. So if you were, and that was coming from breeders, if you look at breeding, will it be more interesting to know that the variety is high protein in the seed or high protein in the meal?
It is more important to know high protein in the meal. So let's say if you're having a high efficiency in oil removal, you still can end up with high protein in your meal. And in the end, that's our start material for protein processing. So that's the most relevant one. But I think there's always kind of coupling between the two of them.
Because actually that was one of the difference between the two processing, your processing at DSM and the processing that they had in Canada. You were starting from the meal where they're starting from the seed actually.
Yes, exactly.
So that's a bit of a different process.
Yeah, no, absolutely, absolutely. If you start from the seed, then you also need to valorize the oil in a very good way, and you need to purify it. While in our case, the oil is already gone and it's valorized. So
yeah.
Case. Yeah.
So yeah, it was very interesting, and the taste, because and that comes back to maybe one of the study that Soren did, and what I was mentioning, and maybe Soren can comment on it. piglet and a lot of animals are very sensitive to taste. So your system showed that actually there was almost no taste. And that's also very interesting because you don't add nutty flavor or bitterness or astringency so much as you might add in other system.
Yeah, correct. Now, I must say the rapeseed protein itself has a quite peculiar taste. It is like other proteins, is a bit astringent and also has some bitterness. But the most striking thing is that it has a quite sweet taste, and we call that the licorice taste. But this is if upon formulation into our applications, you can remove that completely. And that's of course very interesting and also important because sweetness as a main taste is sometimes difficult to work with.
And there was, and I'm sorry, there was also a question about processing, and I'm not sure if you can answer it or not, but there was a comment from somebody and kind of a question at the same time that actually if you remove the hull of the seed, then you also decrease the fiber content before crushing. Yeah. And do you know actually if it's a processing like that that will be favorable for your processing then, or is it possible? And I'm not sure I put you on the spot because this is more kind of crushing than really what you're doing, but.
No, I know, no, but of course we have looked into that. And yeah, there's no straight answer. It really depends. It really depends on how well you are able to crush afterwards. Because if you remove the hulls, crushing will not become easier.
Yeah.
The good thing is indeed you have removed your fibers, and therefore the remaining part, the feed value, will increase. So that's attractive. On the other hand, you will also lose kind of your part of your protein in the hulls because you never can make a clean cut between the and the kernels. So yeah, that's the case you need to consider for yourself. I would say. It is in the end less favorable than it seems in first instance.
Okay, thank you. Another question, actually, a lot of questions about processing now.
That's good.
What will be the minimum threshold that you need for protein content and meal protein content to have a good process?
No, I think the winter seeds are the minimum. So these protein levels that we have in Europe, so that is around 18% of protein. Go lower, yeah, the economics become difficult. So you really need to have a high. We look for high protein seeds, of course, because it makes your life easier. If you have lower protein, will it be impossible to process? No, I don't think so. You still can process. But it is clearly, if there's less protein per kilo meal, also there's less protein to extract.
Okay, thank you. And last question, but not the least, what will be one of the anti-nutrient that should be really eliminated for your process? If there was one anti-nutrient that you have to choose in the canola seed to eliminate by breeding, which one will it be?
Yeah, I think that is the polyphenols might be the thing to remove here, because they are really nasty ingredients, and they are not so much anti-nutritional factors, because they can be quite healthy. In fact, in tea and coffee you consume them as well. But the thing is that the polyphenols, they bind to your protein during processing. So if you by breeding already lower these levels, that might be very helpful. The other things, like for instance, the glucosinolase, so the mustard taste, that can be removed. And also phytate levels can also be removed. The polyphenols is the most difficult one.
I have to say that this is really bad that you cannot use our canola because we have over 22% protein very often. And there is some variety with 5 micromole per gram of glucosinolate. Maximum is 10 to 12 in the meal. So that will be very nice.
Yeah, absolutely. Yeah.
Too bad.
Yeah, I cannot change it in Europe.
No. So thank you. Soren, I have a question for you. Your experiments show that actually you cannot really replace meal by rapeseed. The piglet didn't eat as much. They didn't process the food as much when there was rapeseed. in as a replacement. Do you think that was some of the component on the protein that you give them? Do you think it's something that was impacting the digestion, the taste, or whatever in your experiment?
First of all, I will say that it's a very tough challenge. We put both the rapeseed and the rice protein on to substitute a milk protein. So, the experiment was both related, for the piglets, but also related what will happen if we want to substitute milk protein in human diets. So it was both a model study and it was, of course, the piglet study by itself. But if we look at the in vitro digestibility in the very last part of the presentation, it shows that we have a little bit lower speed of digestibility of the protein. So I think we can have three explanations for this little bit lower feed consumption and feed utilization when we include both rice and rapeseed, and also just rice and milk protein. And it is, of course, there can be something with taste, and it has already been discussed with the DSM technique. And so it can be the polyphenols, it can be glucosinolate residues, but it can also be that the fiber, they take up place in the digestive tract, so you lower the emptying of the passage rate of the digestive tract, so there's simply not space in these small piglet stomach in order to take enough feed up over the short time they have available. And then finally, also what we could see with the lowest digestibility of the protein isolate, we have not knowledge here about whether it is mainly napine or whether it's cruciferine, but looking at the structure of napine and cruciferine protein, we know that the napine contains a lot of sulfur-containing amino acid, a lot of cysteine, and they have these bridges between the cysteine, which we know from other cases that they can be difficult to digest. So I think there are 3-4 likely explanations for this small lower, relatively little bit lower feed consumption.
No, it was a tough one. And do you think that all the animals, like if you did another experiment with, will be the same for when you give the milk, to replace milk for other animals, will be the same issue?
I think the piglets they are more challenged because they have both limited feed intake capacity, they have limited amount of digestive enzymes, both for the different proteases and also other digestive enzyme, and very short transit time in the small intestine. So they don't have a long time to digest the feed. So I think the piglets, they are more susceptible for small changes in the feed composition than more grown-up animals, where you can have more time for the digestion.
So you really make your life very difficult with that project.
Yeah, and that was the aim of it, because we should challenge it.
Yes, but I thought that was very interesting. And Wolfgang has a question too.
Yeah, I wanted to change topic actually.
Go ahead.
Amin Abadi has talked at the end of his presentation about breeding, of course, and genomic selection, and then he has presented a stepwise procedure. Number 1 would be increased protein content. Number 2, then deal with the type, cruciferin and other type. And third would be to reduce fibrate glucosinolates, for example. So the pre-requirement would be to have a certain minimum protein content before you go into other details. What would be the requirement, you think? What would be needed, and what can be achieved in rapeseed with regard to protein content?
I think that the question is directed to me.
Yes, please.
Okay. I showed that in my presentation there is a large genetic diversity for protein content, at least not in the released cultivars in the last 5 years, but in pre-breeding population and so on. So I suppose that 20% to 22% protein content is achievable, but it will be a long way down to achieve that. And the second thing is that when trying to look at the protein content, I think it was we have to look perhaps then at the protein yield and not really then at the protein content itself, because at the end of the day, what you are harvesting is that what you're getting in the seeds. And so so this is a really much complex things. Then you need yield, you need insect resistance, you need the pathogen resistance, you need and then you need then also protein. So we have to do it like we did with the oil content, and then much more concentrating perhaps then on on protein yield.
Thank you.
Actually, I mean, since you're on the spot, and maybe Jung-Soo can also answer, because the two of you are geneticists and brilliant. How difficult will it be? Like Gergent mentioned polyphenols, reducing polyphenols, and it was step number 3 in your process. So what will be the time, and how to reduce those components that actually are not that big, you know, small components, small amounts. But and in your presentation, actually, the polyphenol was one of the problem giving the bitter taste and the astringency. So that's one of the one that you have to remove.
But perhaps just another question to smallers. Which kind of polyphenols should we reduce? Because the family of polyphenols is quite high, and you have a large diversity there. And the pathway is for the metabolic pathway is still under study, because you have some enzymes then where you have perhaps then to tune it a little bit, but we still need the final product that we should eliminate, and then in the breeding. So if we know that, and then with the new tools that we do have then in genomics, then and also in reverse genetics, looking, because it should be the non-GMO, we should look for mutants then for that, or looking in our breeding material for that. I think it will be a little bit easier to go a step forward in this direction. I would like to invite you to look at one paper that has been just released, I think last week, from the lab of Bernd Weishar, where he describes the genes that I have had then in my presentation for the K3OSS and then on the polyphenol pathways. And in Brassica, or in Brassica napus, you have a family of 13 genes for just one enzyme. So for breeding, if we go for breeding and in combination with other traits, then you may know how it should take then to get the right compound there afterwards.
Yeah, I think indeed doing selection both on polyphenols and also on protein at the same time will be very, very difficult and take very long time. Starting with the polyphenols, I know, for instance, the work that has been done by the group of Hofmann. He has shown that camphorol equivalents were one of the main components for bitterness. And I know that is indeed one of the components that is a problem, but it is not the only one. And I think that is the issue with the polyphenols. It is a very, very broad family. And the obvious ones, like sinapin, for instance, that's not the main problem. That's much more into the smaller polyphenol quantities that create this problem. But what we can't tell you at the moment. So this really needs to be investigated more closely to see which one is really the issue. And it's not only the component that was assigned by the group of Hofmann. But I will read the publication if you send the reference to me. But I expect that it is much more complicated and you need to address multiple things at the same time.
Yeah.
Unfortunately.
But at least just to say then for the taste and for this compound that is producing this astringent taste. So we know a little bit then about the enzymes that are involved in, but still it is a tiny compound in the in the concentration. And it must be some things that perhaps then you can eliminate by processing. I don't know. Yeah. It would be better to look for perhaps then processing methods than how to eliminate that than breeding, because it takes a long time for that.
No, I understand. And if there would be one component or maybe a few that you could point at as the problem makers, then you could indeed say, I'm going to develop something to remove, although that might not, because these in extraction you have complex mixtures and it's not so easy to isolate one component only. The other thing is that during your extraction, the polyphenols are not stable, but they are active. So they other components, and you are building slowly intermediates that are even worse. But to analyze them is very difficult because they are not stable. It's a moving target, so you can say. I think in general, reducing polyphenol levels will help, but it is not guaranteed that you will solve the problem.
Even worse, that these compounds can also have some interaction with the protein fractions, and so now they bind to the protein.
Absolutely.
And then you cannot remove them afterwards.
No,
this is the big problem.
Exactly. So if you could somehow avoid that, then you would also be in a much better state. But as far as we know, it's a kind of natural behavior of these polyphenols that they are able to. They bind to your protein, and once you allow that, there's no way back. So it is a product. We have also spent in our processing a lot of time to properly remove polyphenols. We came a long way, but still there's clearly room for improvement.
Thank you. Wolfgang, you have a question?
Additional question to Jan Smulders. Obviously, polyphenolics are important during processing, of course, since they bind to the protein. But in the final product, in the isolate, you have what, 95% protein, and the residual would only be 5%. Would you see variation in the residual with regard to different compounds?
You mean in terms of polyphenols?
Polyphenols or whatever.
No, the process in our case is pretty stable. So let's say the product composition that we have is more or less stable. We don't see variation, but we have also, it's not something that we do on a regular basis to check for all the polyphenol classes because they are very difficult to measure. You get a lot of people.
Yeah. Thanks.
Oh.
Amin, you have a question? Go ahead.
Yes, again to Gertjan. Did you try different genotypes than of rapeseed, for example, for the yellow seeded rapeseed or something like that, because polyphenols are also a major fraction than in the hull in the coat of the seeds. So is that an alternative then?
Could be. Of course, we have tested the varieties that we could get in Europe. Also, we looked into varieties that we could get from Canada. But we mostly were focusing back then on protein levels because that's the main driver for our process. But in the next stage, it might be interesting to look if you can influence indeed polyphenol levels by working with different varieties. If there are some that are really different, that's interesting to know.
So I think the message for us then for breeders, we have to go for protein content, at least then for the protein in the meal fraction. Is that correct?
That's correct. Yeah.
Okay. First, and then the other things.
Absolutely. Absolutely. In that order, I would recommend to do that.
So actually I had the question from the audience, and that was actually one of the question or one of the comment that dairy cow, and we use a lot of canola meal in Canada for dairy cow. And there was no mention of protein used for dairy cow and ruminant. So maybe actually there was, Soren, maybe you can answer that question. And then also Jimmy and Richard from Australia, you had you were working with ruminant and protein in ruminant. It was feed cow, but what will be about dairy cow?
I think here at least in Denmark, and I think most part of Europe, the major part of the rapeseed meal is going to ruminants, to dairy cows. And there are both good things and bad things about that, because in Denmark, and I think also in other part of Europe where we have regulation with phosphate levels in the manure, there are maybe problems by including too much rapeseed protein, rapeseed meal, because there's simply too much phosphate, and we have most of it is phytate bounded. So that's one problem. But then on the other hand, what there is, in order to reduce methane emissions from the dairy cow, it's been done quite a lot of research here in Denmark about both with press cake from rapeseed in order to increase the oil content, but also with crossed rapeseed with the complete oil content in order, as they can reduce the microbial fermentation in the rumen and thereby decreasing the methane emissions from the cow. So there's it's both and, I will say. That's from my point of view.
Richard or Jimmy, any comment from Australia? Because actually there was also a comment in your presentation, Richard, from that there was a reduction of methane using canola meal and canola oil. And there was some work done on it in Australia.
Yeah, absolutely, Veronique. So, I mean, there's a fair bit of use for canola meal into the dairy industries in Australia. I think I had 25% there. So I didn't. I suppose the research I focused on was around grass-fed cattle, and some of the research that was in that area. But certainly dairies, you know, a very big end user of canola meal in Australia. As far as the methane work goes, it's there has been some work done in Australia Obviously, Roger Hegarty has done a fair bit of work with canola meal, and I think cold press canola meal with a higher oil content. So there's a few things going on there. I suppose a few mechanisms. The high protein content of canola can increase the efficiency of digestion of some of those roughage feeds. I'm thinking again about grass-fed livestock, but the same goes for dairy as well. And the other mechanism is the oil content can directly inhibit methanogens as well and reduce methane abatement there as well. So it's a continued, obviously there's a fair bit of work and there'll be continued work going into that area going forward. So I think it's a pretty exciting area of development for canola meal into the future.
Thank you. And sorry, Zhangju, I saw that you have your hand raised and I miss you. So if you don't mind, sorry about that.
I want to, I have, there is a point from the presentation from Richard Mell. There is identification of the cultivars with high oil content and protein content. From my understanding, there is a very significant negative correlation between the protein content and oil content. So is there idea to select the cultivars with both high oil content or just with one direction? Because for our breeding cycles from the past to now, the seed oil content has been increased largely. At the same time, the protein content is in the is very negatively with the oil content. So to breed the cultivars with both high oil content and protein content is possible, or do we need some balance between these two traits, or which direction in the future for the protein content? Or we just focus on select cultivars with high oil content and also the protein yield, high protein yield, or what kind of breeding targets for the protein content for considering the correlationship with other traits?
Yeah, absolutely. I mean, that's a fairly strong correlation on that graph, and obviously across all those varieties that we looked at. And while we did identify a bit of an outlier group, I suppose, that seemed to have consistently higher protein, higher oil content, that was something that probably can be explored into the future. And Jamie might be able to cut in and maybe make a bit more of a comment about that. I think he's on deck.
Yeah, can you hear me? Yeah, I think you've covered it there, Richard. There was that outlying group that we found that had relatively high oil content and protein, and we'll look into that further in the future. But yeah, that's, as Richard said, there's a lot of work to be done, so we'll continue to do that.
I think the interesting part about that too, Jamie, was those 3 or 4 varieties that we looked at in there were fairly consistent across a couple of years, and across those sites there was fair diverse climate across those sites and pretty consistent results. So there might be something interesting to go forward with there as well.
Yeah, I can't add any more to that, Richard. Thank you.
That's fine.
Thank you. Actually, we have somebody from the audience that would like to ask a question to the panel. So Friedrich, please You can ask your.
Just a moment. Was my technique here? Yes. Thank you for the nice conference via Zoom. Sören, hello. Sören, I'm very impressed by your piglet experiment. However, you used a unique model. That means the most sensitive animal, and you used it in the very sensitive phase, yes. And we now we say, or we give minimums or limits, yes, for the piglets, for milk protein, for milk products in the diet. And my question is, maybe you have a good, too good control for this comparison, because we have a unique digestion process also for the milk protein. Yes, remember the chymosin hydrolysis in the stomach. It is unique for the milk protein, and I cannot understand that we have another protein which is similar to this milk protein. Yes, these are mammals. This is my, maybe it's only a comment, and my question is, what about, you have protein isolates, and I think a protein isolate is fiber free, and I was a little surprised that you have a fiber content in this isolate. And what is the nature of this fiber? Is it cellulose? No, yes.
Yeah. Thank you, Friedrich. Yeah, tough questions you asked, but yeah, I mean, I think, first of all, you can always, when you have a protein content of 90%, 85%, or even 95%, how the first question you can ask is how do you calculate the protein content? If it is just is n multiplied with 6.25, then that's not appropriate way to do it when you have such a high protein content. You need to count the amino acids and then see if you have any residual nitrogen. But in the protein isolate, you'll mainly, all the nitrogen will belong to amino acids. And there was a small discrepancy between in both in the rice protein and in the rapeseed protein we used whether we simply measured n multiplied with 6.25 or 5.75 or calculated the amino acids. So we used the amino acid content, and then there is, with respect to the fiber, you can ask where do they come from. I think it has something to do with the way, I'm not completely familiar with the way that made this Toitex protein isolate, but I think it's, even though that you make it mainly from precipitation, you will still have some fiber bound, maybe via some of the polyphenols together with the protein. So what we measured was mainly some lignin. I can't remember it right now whether we had cellulose, but I think we did complete fiber analysis on it. But there was a little bit higher content of fiber in the rapeseed than there was in the rice. And I think that makes sense that you have, because it was also darker in the color. And again, we're back to the polyphenols or lignin. So yeah, and with respect to digestibility, I think it's simply different mechanisms whether you have milk protein, and you know what we are searching for is something which can substitute milk protein, but it may do it in another way than we digest the milk protein. So I think that is what happened in the piglets.
Thanks. Okay.
Thank you. I have another question on the chat, and it's regarding the processing. And one of the comments was when you do the processing, you end up with a lot of water. And how do you get rid of the water, and then the cost for that? and the energy to remove the water. So this is also on processing for Gertgent.
Yeah, so the question is how to get rid of the water?
The energy, because you do probably a water extraction system, and then you have a lot of water to get rid of, and the energy removed to get rid of the water, you know, how efficient is it, or how difficult is it?
In general, you use ultrafiltration to get rid of the impurities that are part of the rapeseed. And that's also the way how you concentrate your protein before spray drying. That creates still a lot of side stream water, but we do recycle that as much as possible. And we also try to use as much as possible the energy that is in the water. So you have heat exchanges, etc. So we try to optimize and reuse everything, because there's a lot of water going around in such a process. If you don't deal with that correctly, then the cost will be very high. You're muted.
Was also coming back to you again. I think it's so interesting and so new to have this big processing and commercial processing, actually, that we know is going to be in commercial product and going to hit the market. It's so interesting. So there was a comment that there is some, because you say it was 30-40, but there is something that for cruciferine and napin, but if you have only 20% of napin, how is it going to affect your product? Because there is some variety that's much lower in napin than the 40%.
Yeah, I think if you have a crop with only 20% napin, first of all, we would not use it in our process right now, because we want to have a certain quality, and we target, of course, to be stable and use one kind of rapeseed for that. Nevertheless, if you are having a different target, you can, of course, use a crop with a lot lower napin and for instance, higher cruciferins. So you can make choices there and have different product qualities. That's something that will happen in future. I'm quite convinced about that. But right now, where it's a new product, new process, we need to get into the market. So you come with something that is, the best option right now for application and further development.
Thank you. And Soren, I see you have your hand raised.
Yeah, and that's also a question about processing. I just wonder how, when you have the change in cruciferine and napin amount, and how so it lead to the question how big proportion of the total protein in the meal ends up in the protein isolate you are making.
We do not disclose that, because that's kind of disclosure of your processing yield. That's, let's say, confidential information.
Yeah, but then you can calculate a little bit about it. But when you have the napin content proportion in the meal and what it ends up with in the, so it seems like you lose quite a lot of cruciferine. So I can maybe ask another way: what do you do with the remaining protein?
Yeah, we have we do not extract all the protein from the material from press cake. That's impossible also, because there's always part that is just bound and you cannot get it out. So we have, again, a side stream in our process that is creating a wet cake, as we call it, and that is also a source of feed again. And that is not completely zero in protein. So there's still protein in there.
Yeah, I'm just simply just in, as we're discussing a lot these days about sustainability and then proper utilization of all different kind of side streams, and just important to see the whole way around to utilize all the fractions.
Yeah, now we have tried to utilize the maximum that we can utilize, and we are not at the end of it. It's the first plant that we are building, and we think that for the future there will be much more possible, but then you also need to first see that the business is there and that it develops. But in the process that we are building, for instance, all the wastewater is treated in a wastewater plant. And of course, this is converted through anaerobic fermentation to biogas. So also there you utilize what is in the water totally to the max, because it is converted back to gas that we deliver back in the system.
Thank you.
Yeah.
I mean, you have your hand raised.
Yeah, Gertjan, excuse me, I have to I have to ask another question then about cruciferine and napping. So we know from the breeding sites that then if we have to raise the ratio, let's say napping, or lower the ratio of cruciferine to napping, we will have also some problems then with glucosinolates, because there is a linkage drag between the napping fraction and also the glucosinolate. So is that a kind of compromise that you can deal then with glucosinolate afterwards? Because there are some old varieties that are higher in napping already.
Yeah. I think we can deal with that, because the glucosinolates are not so much of the problem in the process. You can remove them relatively well. So even if you have slightly higher concentrations, I don't think that it will give you big issues. Mustard, that's really a different kind. That's much higher in glucosinolate levels. It can be, I think, factor 2 or 3 higher. So that's
what does it mean then much higher? What is the concentration then for you in the seeds, for example, then for glucosinolate that might be acceptable then for your process?
The normal concentration that you have now in rapeseed, that is what we, and in the canola, that is what we can process, and we do not experience taste deviations in our end product with these concentrations.
Because the normal one, which we do have in Europe, is quite about 18 micromoles then per gram, and then
I think 20
in Canada you do
lower,
have then 12.
Sorry?
In Canada is about 12 micromoles then per gram.
Yeah. So I think in the spec sheet that I had in my presentation, it said 12, 20. No, that was the end concentration. But I think in the rapeseed that we use, sometimes it is up to 20, and that is what you can deal with. Yeah. But must be clearly higher.
And Etienne, you have a question?
Yes, it's a complementary question. In fact, when you say that you can manage the glucosinolates in the process, does it mean that you leave it in the cake, or you extract glucosinolate out of all the products?
No, glucosinolates are also extracted, so they are soluble and they come into your water phase, unfortunately.
Yeah. Thank you.
Well, unfortunately, or maybe not, because actually there is some beneficial effect. There is some health effect of the glucosinolate, and also I know that you can use it for something else. So actually, you might find in your process later on something to do with the glucosinolate that you extracted, because actually they're in almost pure form. So that might also be interesting. And as an analytical chemist that analyzing glucosinolate, I'm always very interested in finding reference material and glucosinolate extract. So, you know, a lot of people like me will be very happy to get your glucosinolate.
Yeah, no, absolutely. You are fully right. The glucosinolates are, of course, very interesting components that you can extract as well and isolate as well. The same for the polyphenols. So the polyphenols that you extract can also be isolated and sold as a separate product, and it will have very specific properties that might be of interest. Absolutely. But for I think that's really the future. I'm sure that it will go into that direction, but not in the coming years, because you first need to get the plant up and running and sell the protein. And if that happens, then you can take the next, go to the next generation. Yeah.
Actually, I have another question, because we have Brassica gensea. We have Canola Brassica gensea, and I don't know if you have Canola Brassica gensea in Europe, but he has yellow seed coat, and it's still, he has the quality of a canola. So have you ever tried it in your process, or you just look at Brassica napus?
Yeah, we only looked at Brassica napus. But I'm not so sure if the difference in hull will affect the end product quality in the end. But that needs to be tested, and I don't know.
Okay, thanks.
Yeah. Okay.
And actually, I have a question that might be interesting for the maybe Rob and Jimmy and from Australia can answer it. And there was a question about the severe drought that we have this year in Canada. So we were experienced very high temperature and then very low moisture. And there was a comment that moisture is, and high temperature is affecting oil and protein. And I can say, yes, oil content is much lower this year in Canada that we have for the moment. I think we've been analyzing close to 1,000 samples, and we're about 41% oil content, which is about 4% lower than normal. So a comment from the people, how do you think like this year, if you had the model, Rob, how do you think this year, this growing condition this year will be affecting your model and the oil content and the protein?
That's a great question. Now, I'll preface that with, while this year was extremely dry, we're actually certainly in parts of Western Canada in our 4th dry year in a row now. So this isn't, I think the big difference this year was the high temperatures. I mean, we've had relatively low precipitation, and certainly below normal for the last 4 years. You know, the data that we've got for the from Corteva that ranges from 2017 to 2020, I would say there are some relatively dry environments in that already. But absolutely, this year will be different. I'm curious to see whether we see, if we were to include this year's data, whether we see an increase in the year by environment, in the year interaction, because the year effect throughout this analysis and the interactions with year have been relatively small in terms of the amount of variation that is partitioned off to the year effect. So the range of environments that we have across Western Canada still trumps whatever variation we have from year to year. But now that we have a more extreme year, that may change. I expect to see the variance partitioning towards the year effect increase. How much? I don't know yet. It's a very good question, and I'm curious to see. I would also like that at the same time, with a number of our studies that we've been doing over the last few years, would like to see a wet year again, so that we see what happens in our what happens agronomically under more normal conditions. But it's been quite a while in Western Canada, certainly where I'm located, that we've been working with below normal precipitation.
And comment from Australia, Jimmy, maybe, because I mean, I know you have a dry year in Australia. I mean, it's been wet, and then because of that, you have a lot more canola in the last 2 years that you ever had. But before that, it was very, very dry, and the same condition that you have hot and dry, we had. You also have them in Australia. So, any comment for that?
Yeah, that's correct. We, as Richard said in the presentation, we're expecting record yields this year because we've just across the board, we've had a fairly good year. I'm an analytical chemist, so I don't want to go too far out of my comfort zone with talking about agronomy. But yeah, we won't harvest until later this year, so we're thinking that yields and oil contents will be higher because of the mild autumn and winter that we've had and the rain. But as you said, Veronique, the last couple of years before that, so 2018, 19, were very dry, and we did see the effects that you guys are seeing in Canada now, with the lower yields, the higher protein, and the lower oil.
Yeah, that's what we're seeing in our sample this year. As I say, quite lower oil. And we never talk actually, I know this is a protein session, we're not talking about fatty acid, but I know that the fatty acid was also affected by the heat this year. So a lot more in saturation, more saturation in the oil when you look at oil quality. And actually, I'm sorry to say, yes, it's a protein session, but oil is very important in canola. So oil was also affected by the heat this year in Canada. So any more questions? So the people from the audience, do we have any question from the audience? We can unmute anybody, so it will be nice if we have a question from the audience. So Friedrich, go ahead.
To Richard Meyer from Australia. The feeding pattern in Australia is very interesting and completely different from Germany. You have a dominance in the feeding for the poultry, yes, broiler chicken, and then we have an impressive part for the pigs. And in Germany, we have a dominance of the cattle sector. We have it heard. Also due to the GMO requirement, and it is GMO-free rapeseed here, yes. Okay, and my question is, I know Australia is also the world champion in lupins, producing of lupins, yes, lupins, a legume. And what about the feeding practice? Is it a combination of rapeseed meal with the lupins, which dominates, or is it a sole feeding of rapeseed meal? What about the experiences in Australia?
Yeah, thanks for the question, Frederick. Australia, we do tend to, so I guess the biggest protein meal that's used in animal industries is probably, like, there's a fair bit of cottonseed meal that's used, and cotton is yeah, it's become a lot bigger, especially in our part of the world, down in southern New South Wales. So that's used as a fair supplement. Canola meal obviously is gaining popularity. And there are some other meals to a lesser extent. Yeah, so I guess and look, there's quite a lot of extensive livestock that don't do a great deal of supplement with meals at all, that do. So there is a reasonable amount that use lupins and use other sources of protein, I suppose, like lucerne and actually during our drought periods over here, there was a lot of recovery of canola crops that would have gone on for oilseed production that were actually diverted into canola hay and canola silage, and that's actually gained a fair bit of popularity, like even outside those traditional drought periods. There's still a reasonable amount of production, believe it or not, of which won't probably happen this year because of the price of the high price of oil seeds, but it had become popular where there was production of silage and canola hay that was outside the like a drought recovery phase, I suppose, as a protein supplementation to just normal forage.
Yes,
I hope that's answered your question.
Interested in this combination effect. That means peas and rapeseed meal, or seed lupins, lupin seed with rapeseed meal, yes, and also the faba beans, yes, very interesting in the amino acid combination, yes. Okay, thank you for this.
Thanks.
I might have a question actually for the breeding people. There was a comment from Amin at one point, and it was long ago that I think it was Balder Stevenson that said that you can increase by 1% a year. So in the condition that we have now, where we need to breed for diseases, we need to breed for insect, we need to breed for heal, how much do you think an increase could be expected in a year? You know, like will be 0.2%, 0.5%, 1%. What do you think could be done?
I think there was a small mistake then in the reproduction of the statements then in the workshop, because it was written like this: because 1% protein increase, 1% protein increase in a year, it's not possible. I think it should be 1% protein yield in a year. It could be possible.
I was thinking actually, because one of the analysis was done with Tobin and Tower, which is, you know, like right at the beginning of the canola. And I was thinking, well, at the time you could breed for quality because there was not a lot of disease and resistance. And now I mean, if you look in Canada, you're looking for diseases, you're looking for pod shattering resistance, you're looking for heal, you're looking for all those things. And I was thinking that the increase in the quality that you want to have has to be a lot smaller than the 1%. I was very surprised to see 1%, but I was thinking, well, it was 40 years ago, you know, in 1976. So it was early beginning of the canola.
Okay.
So do we have any more questions in the audience? A couple of last questions, because I think we're supposed to finish in 8:30. So we have one question in the attendees, so if we can unmute the attendees, please, and then I have the question. And somebody has a raised hand in the attendees. Okay, then Duran. So actually, Etienne, you have also a question?
Yes, in fact, I mean, looking at the presentation by Amin yesterday, there was a lot of historical things, among them this assertion of Balder Stevenson concerning the raising the issue of breeding for both oil and protein at the same time. So my question, in fact, is for breeding orientations, do we have to select today for proteins alone, for oil, or for protein and oil? And does it affect the way to characterize varieties, for example? I mean, if we choose a global criteria oil plus protein, it would mean that in some way we leave the adjustment of a ratio to agronomy and environmental effects, in fact. What do you think about this?
No, I think at the end of my talk, I was also talking about sacrifices, and at least then we need also a legal frame. That's what you are pointing out. So for registration of the varieties and so on. So that means then what we hope as breeders then to have is a really a roadmap, what we have to do in the next 10 years. Because just yet trying to say, okay, we have to raise the protein content and we have to let the oil content at the same time, it's not possible. So we need a certain direction where we have to go, and then this has to be then at least then perhaps then discussed a bigger frame, and in Europe, and also then at least in Canada is they already have a cutoff. They have a kill criteria for the registration. You have to have a certain protein content in the meal. So otherwise you will not have the you will need but be registered. But we don't have it in Europe. And the second thing is then whether there will be also a premium on that. If we go for protein, whether the industry can also have give them a premium on that, because at least then is an investment. There's a big investment that you have to do as a breeder, and then also the farmers then should have also some benefit from that.
Thank you.
Actually, that was a decision that was done in Canada a couple of years ago, that if you want to have the producer, you need to have the breeding. So there is target. You cannot be so lower than that for oil. You cannot be lower than that for protein. You cannot be much than 12 for glucosinolate. And you cannot be more than 7 for saturated fatty acid, for example. So that's some of the parameters that you need to have to be able to register a new variety in Canada. And that's on top of the yield and the disease resistance and everything. So quality there. There is somebody from the audience that had a question, and please go ahead.
Thank you, Veronique. I will broaden a little bit the question from Etienne. We have different possibilities to be not only oil versus protein, but it seems to be important that the different fractions of the protein, like cruciferine versus napping content, or looking at the different anti-nutritionals, glucosinolates, polyphenols, the beta taste compounds. So the spectrum is getting more complex. How to build a strategy? Different colleagues, you had different opinions. What should be more important? So how to build a strategy for the breeders and for the future developments? Thank you.
That's actually the $1,000 or $1 million question, because I have to say the last time I talked to a kosher, he told me my best canola ever is just oil, 99% of oil, and just a membrane around it. That's it. So we in the protein session here, so I expect a different answer than that. But and I think you just target the dilemma of the canola breeding. We discover now that there is so many things that we can do with that seed, because that seed is such an interesting seed for the protein content, for so many things that we have, and we need to do more than just the oil, because for many years I know in Canada the oil was the top, and then the meal was kind of whatever. And now we find actually there is value in the meal, and there is value in the whole seed, and so yeah, so that I think this is actually the question that probably going to summarize all this session. What to breed for? Is it what it is? We know it's the oil, and we know it's the oil because at the end of the day it's a big product, and that's 40% of the seed, or 50% in the seed in some case, but I guess the other things are also very important, and it will depend on the audience, I guess. So yeah, I don't know. And
probably on the use of the products you're going to produce.
Yes. And that was actually, and maybe this is part of the discussion, and further discussion is could it be some we call that IP system, protected system, when you have somebody that's growing a small quantity but sell directly to a processor, because that processor is doing exactly that product. And whereas in Canada we have this system when it's protected. identity protected, we call it IP. And then we have the general system where it goes with the normal way. And that may be one of the way, I don't know, might be one of the future of the canola industry, that you have many different small product and small target breeding and different company breeding for different things instead of breeding for the same. But I think that Wolfgang and Amin are going to kind of talk about it. And they have the hand raised, both of them, so go ahead.
As to priorities, I would say it is good to have different usages, because this would also be a pre-requirement for a certain diversity in varieties which we have in use. The genetic diversity is still already very small, and we have to keep this on a as much as high possible level. And we need to really extend diversity, because as we have seen clearly yesterday, we'll have serious problems with pests and diseases and environment stress. So we need diversity, and this could also be reflected on the level of use. I mean, would be a basis for genetic diversity on the field.
Veronica, I wanted just to comment on the IP system. Then we, in Europe, they also are familiar with this system. They're using that, for example, then for the high-acidic acid rapeseed. So this is a closed system. Perhaps then for first, it could be a solution, or for the high protein.
Yeah, we have IP system for the high-acidic and also for the high protein from Dow is an IP system. So they could be different treatment, and you know I know Cargill, as for example, they're going to come with the high PDHA canola. There'll also be an IP system. So canola breeding is wide and generous, and actually this is probably because it's a genome that's been well, you know, there is a lot of knowledge on the genome, so it's a lot easy to do something out of it. But yeah, I'm not sure we answer your question because there is so many things that we can look for, and then we could be, you know, people could be very demanding.
And I assume that each one of us has some different opinion depending on the downstream products we want to produce. But it's a good starting point for a discussion. Thank you.
You're very welcome. Thank you. I have a question from Jiangsu, actually, because China, it's a very big production for rapeseed. And I know that some of the quality in China could be quite different for the rapeseed. So where do you see the rapeseed quality and going in China? How do you see it going? And maybe if there is some Chinese colleague, and they might be also able to comment, raise your hand and comment. I saw that Dr. Purley is on the panel, so maybe I also can comment on that. How is it going in China for the quality of the rapeseed?
Most of the cultivars, the open pollinated and the hybrid cultivars is a double null seed quality. And the breeders and the market more focus on the high oil content, high yield, and we also have some market for the high orninic acid content for the oil. And in China market for rapeseed production. We grow in rapeseed. The major part is for the oil with this double no, high seed oil content, high vanillic acid content. And on the same time, some rapeseed was growing for traveling and for feed crop for animal feed. And there's some breeders also interested in for the breeding with the protein because the soybean is also, we need a lot of protein, and I think it's a very maybe it has a huge potential for rapeseed as the one source of the protein. But this is very just the start. We didn't have a lot of work on the protein, and also found is the protein content. This trait is very complex, similar or even more difficult at the oil content, and also the processing of protein content. There's a lot of technique or questions need to be resolved for the processing of the tasty and the content in the protein. So yeah, so one point is more focus on the high oil yield, and also for some functional nutrition for the rapeseed, and yeah.
I have another question. You mentioned that you have open pollinated and hybrid. Is it a majority of open pollinated still, or a majority of hybrid, or different, you know, like if you had the small farm, you're going to have open pollinated, and if you have big farm, you're going to have hybrid? How is it working exactly?
Most of the cultivars in the Yangtze River, Yangtze River is the major rapeseed production area, and we're growing hybrids. Most of the market, the hybrids occupy the most of the markets.
Okay. Thank you. Thanks. I've been in Wuhan a couple of times, and I saw a lot of the field around Wuhan. So interesting. Thank you. So actually, I have a question, or I have a comment. I think the DSM system is going into human nutrition. And when we had the Canadian system, and it's too bad Dave is on the meeting, but I know he was traveling, was going more into aquaculture and human animal nutrition. So what do you think will be the part of the human nutrition versus the animal nutrition for those protein concentrated? Or will it depend on when you're sourcing, like in Canada, because GM will be more into animal nutrition, whereas the European seed will be more into animal nutrition? How do you see it? Okay. So actually, I have Ivona that has a question into the chat, and maybe we can unmute her, and she will ask the question.
Okay, I have the question maybe for everybody. How long is the perspective we will use rapeseed protein in the human nutrition? Did you have?
Well, it seems like the DSM system, they were going to use it for ice cream. So that's human nutrition, but Gertjan can answer that way better.
Yeah, but I did not fully understand your question. What can you repeat it?
How long is the perspective we will use rapeseed protein in human nutrition? in 5 years, 10 years or longer time?
Yeah, we think that there's a very good perspective for the protein in human nutrition, and the main reason for that is that the nutritional value of the protein is very high. It will take time to grow and also to get into the market, but we think that once we get into this protein in food, that it will grow over years and years, and it will mature in the end. But the processing of the protein is not very simple. That's also, I think, why it has been taken a long time before it happened. Because if you look back in the 80s already, there were stories and publications about that the protein in rapeseed was very good, very healthy, of a high quality. So it is well known that this protein is very valuable. But to get it into a product that is good enough and also economically viable for the food market, that's another thing. But we think, and that's also why DSM is investing in a full-scale plant, that this will start from now on, and that it has a very good future.
Thank you.
I know sometime actually that in the food industry, they're very reluctant at changing the process because they know how the protein are working and they know how the system is working, and they're very reluctant sometimes are getting from, adding something or changing the process. Because if you add it to ice cream, you might need to change your process. So that may be also something that might slow down the adoption of rapeseed protein.
Yeah, no, that's absolutely true. If you are directly competing with already existing proteins, yeah, people will not change very rapidly, unless there are clearly advantages with your protein. The advantages might be in that the functionality, for instance, of the canola protein is such that other plant protein cannot deliver. But then it's a clear advantage. Or that, for instance, in sports nutrition or elderly nutrition, the nutritional value is key, and then you have a better proposition. So there it might be relative fast. So these, and I think you should also realize that the protein market as a total, the plant protein market, is growing tremendously. So even while the current protein products are maintained, still the growth is such that it will facilitate a lot of new products.
And actually, you were also commenting that you were looking for high value product. And if you're looking in sport nutrition or senior nutrition, this is a high value product.
Yeah.
And sport nutrition being kind of new product, then the formulation has to be, you can set up the formulation right away with the system that you have right away.
Yeah. This is exactly as what you do with development of electrical cars. You start in a attractive segment, of course, and from there you roll it out.
Yes, and I see that Amin has a question also. Please go.
Gertjan, this is a very nice example for the electric cars. So you said it also that we talk about the protein quality of rapeseed since at least now 30 years or 40 years from now, and nothing, let's say, there is not a big breakthrough in that regarding really the quality, because I think there was no incentives. So my question is then, which incentives then should be in place then from the market to really accelerate development of the protein quality in, at least in Europe or in Canada?
I think the protein quality is already very high. And what you see also is that for a long time, there has been reported in literature relative low values, for instance, of the PDCAs of the canola protein.
Protein content. The quality is good.
The quality,
which incentives?
If you do the processing right, you get to the very high quality that it can be, and that is what will be the road also to market. And what is now your additional question?
So I mean, we know that the protein quality is good, and so on and so on. We're talking about that at least then at each International Rapeseed Congress and so on. But the problem is that there is really not a big breakthrough then in the development really then for high protein content cultivars. And my question is that which incentives coming from the market should be in place to accelerate the development of such cultivars or such
Yeah, I think that that will happen in the coming years. As soon as we enter the market and you see that rapeseed protein is accepted and has specific functions in food products, there will be a demand for more. And I think if there is a premium also for rapeseed with a high protein, then from that demand and gap that you will get, I think there will be a driver. And that will change. But so far, there has not been a protein in the food market. Discussion and talking and development, etc. But no one has commercialized it. And now you see the first development in that direction. And I expect that if this starts, that it will continue. And from this demand, you will drive in the end these initiatives to increase the protein level. But that might still take time, because we will also start relatively small. If you compare to the total food market, what we do is small volume. But I think also, if you look back, for instance, to whey protein, that is also something that has been developing in 30 years. Initially, that was a waste product, and people were really wondering what in heaven's name to do with whey protein. And some even put it into wastewater plants. If you see now that the dairy industry has completely turned around and whey protein is one of their most valuable proteins. That is because they have been over time developing the right applications for this special protein. And you could almost say that sometimes the cheese making, the cheese is the byproduct and the whey protein is the key. So they make the cheese in order to have the whey protein. So it's a complete turnaround. This might also, of course, there's a lot of development needed and some time, but this might also happen for canola, where you can see that today the oil is the main driver. But if applications are really found, the specific applications where the canola can be unique, might be different. And that will, of course, drive these breeding initiatives, because if protein is the more valuable component, of course, you are going to do it different. Does that answer your question?
Okay, thank you.
Ivona, you had another question.
Yes, I have. Which trait of rapeseed influencing the quality of meal is the most interesting for breeders? In which way go breeders now?
For us, we use a standard rapeseed Napus quality. So we do not go into special varieties.
Not now.
Sorry?
Not now.
No, not now.
Not now.
We work with what we currently is available, and for future that might be different, but we don't know really what to select for or ask for right now.
Are breeders interested in yellow-seeded rapeseed, for example? In the development of varieties yellow-seeded?
Yeah, I cannot answer that question. I don't know.
Are breeders interested in development of yellow-seeded varieties? Yellow-seeded, that's mean?
We had yellow seeded, and I can talk about that. We had some yellow seed in Canada, and actually Gerhard Rakoff from AFC at one point developed yellow seeded variety. And I have to say that in Canada, they didn't really pick up. We still have the Brassica napus. There was another variety at one point that was a bit lighter in the hull. And we had a big problem actually when we were doing the analysis, because we were doing a lot of the analysis by NIR, And actually, there was an interaction with the NIR that was making those seeds high chlorophyll. So it was not really interesting for the people, because most of the crushing plants, they have NIR. And then when they were entering the seed, that seed show up as high chlorophyll. Then you have to process it differently. And then you have to do analytical chemistry, wet chemistry, to find the real content, because actually the real content was low, but it was an interaction of the hull with the NIR that was kind of an artifact showing something. And I think that was one of the reasons why, even if Gerhard Rakoff developed that yellow seed for years, a long time ago, and had good property, there was not a lot of pickup in Canada. So most of the canola that we have is brown. Could be yellow seed Napus, but nothing has been going forward really. And yes, they're much lower. The syncot is a bit thinner. The phenolic compounds are lower, but at the same time, if you don't have any production, it's very difficult to test in a processing plant. So I saw that Soren and Etienne had questions. So Soren first, and then Etienne after.
Yeah, it's a part of question and part of a comment. That's, I think, yeah, well, these little bit more than 30 years I've been involved in this. We have had the same discussion and talk about that amino acid composition is so good, and protein quality is so good, and we have even also for 30 years ago made rapeseed protein, which were really high in digestibility and high in protein quality. I did my PhD on aqueous extraction of rapeseed protein. But so I think it's good that now that big commercial companies like DSM has gone into this business, because it will take some money in order to get this to be reality, to use rapeseed protein for human consumption. But I've just raised my hand a little bit about what I still believe is very important, and that is to not only to focus on that relatively small part of the protein which goes into the protein isolate, but the whole chain, the whole system, to use all the side streams. Because what was the problem when I did my PhD for those more than 30 years ago was that a too little proportion of the protein ended up as the high value protein, and it was not able with this small part of protein to pay the whole process equipment. And I just want to focus on that. It's important to focus on this, because otherwise we'll be in the same situation as we have been over the years. We need a protein where we can utilize relatively high part of the protein. Otherwise it will either also not be sustainable. And that should also be another part of the story. So yeah, that's more a comment than it was a question.
Thank you. Actually, I will comment on your comment after. Etienne, you had a question?
Also more a comment than a question. I mean, if we consider the future of plant-based protein food, of course it is a matter of market, but also probably a matter of political orientations. If you look at what happens in Europe today with the Green Deal, of course it is not yet totally active, but the objectives at Horizon 2030 are very, very ambitious. If we consider what we have to decrease greenhouse gas emissions by 50%, which is enormous. I mean, the nutritional aspect and the dietary issue is at the front line, in fact, of this challenge. And from this point of view, the plant-based food is unbeatable if we consider the carbon balance of this food. So I think in the future, anyway, there will be a huge market for these products. Then globally, of course, if we replace feed, or let us say animal meat by protein, plant-based proteins in food, it will decrease, or it should decrease, the feed market. Then we have to consider also these things at global level, because at global level, the demand will remain extremely high. Yeah. But it means that we probably enter in a period of disturbances, let us say. But I think there is a good future really for plant-based proteins. I don't know what is your opinion on this point, maybe Gert-Jan or.
No, I fully agree with you, and I must say that at DSM we stepped into this development from a program that was called Climate Change Induced Innovation, because we saw all these problems, and that was 10 years ago. We saw all the problems that would happen with climate change, and we also saw the population growth towards 2050. Going to almost 10 billion people, needing more food, and then having no land available to grow all this food. So from there, it was pretty clear that protein would be one of the key topics for the future, and you need to innovate in agriculture and in technologies to process. And we deliberately went out and looked, well, are there sources that are under-valorized and that are potentially rich in protein and could be a good source. And we also didn't want to end up with a commodity, so we also were looking to something that is special. And this is why we started with canola. We, by the way, worked also on other crops, but canola was going so well, and it was so clearly on top of this mondial problem of population growth and feeding the world in a different way, that we decided to continue. And internally for DSM, this was also not easy, because still it is quite an investment to take this step. But you see now that the time is completely right, because the plant protein market is booming. There's a lot of demand. So we are very glad that we have made the step, and that we can be commercial next year. And it is really a solution for the future in terms of sustainability and where we need to go.
There is a comment. At the same time, there is high demand for biodiesel. The demand for biodiesel would decline as politics in Europe move to our elimination of diesel-driven engines. So in 20 years, the European market might be different, and it will be more protein than oil, because yes, actually the oil market, the biodiesel market has been pushing those things, and that's a short period for breeding. So Wolfgang, you have your hand up.
Yes, I was just interested in what the trend will be in the future. I mean, we have got concentrates, protein concentrates and protein isolates with different protein contents. The more protein, the more expensive, obviously, the product would be. So what will be the major products in the future? Will they be based on concentrates with, I don't know, 70% protein, or rather isolates with more than 80%? Can that be roughly said what the trend will be in the future?
You want me to answer that?
I think you are the
This is a discussion that is going on for a long time in proteins, in plant proteins, because what we are doing with plant proteins is that we first purify it up to an isolate level in order to get a very high purity that you can use in many applications. And then you see that in these applications, you immediately start adding other ingredients like fibers and some oil, etc. So this raises the question, why did we purify it up to this high level? Is we start diluting it with components that were originally already present. And you see a lot of initiatives going towards concentrates in that respect. Application of a concentrate might be more difficult because it needs to be tailor-made for a specific segment. For instance, if you make a meat alternative, it has different requirements than where you are making an ice cream or a dairy plant-based milk. So that is, with a concentrate, you have less room to play. You cannot put it in every application. But coming to rapeseed and canola specifically, it's not so easy to make a good tasting concentrate. And that is because you need to remove all these, let's say, anti-nutritional factors. You need to get rid of your glucosinolates and the polyphenols in order to come to a good product. And to do that at a protein level of 60–70%, because that's what the concentrate more or less defines, is not so simple. And we have not, there's not a good example yet of a rapeseed concentrate that is acceptable for the food market. For feed, it's different.
Thank you very much.
Okay.
So we have a question from the audience. You're going to be unmuted, please. Rushka.
My name is Stefan Ruschka. I'm from GEA Company. Somebody knows me in this auditorium, I guess. And I totally agree with Gertjan and also with Søren concerns regarding pricing and yield. We know if DSM is starting with cold pressed material, the protein content is about 33% inside the cake. And then we can, or DSM is only recovering the albumin phase, which is about 40% of this. So that then we talk about 15–16% of the total press cake. And that means we reduce the value of the press cake, which can be sold for €350, down to 1/6 of this volume. That means the price at least for the raw material is times 6. That means 350, 700, €2,100 per kilo protein without processing cost. So it's impossible to sell any isolate to such a low price. So we have to look for niche products. And then the challenge is to reduce, produce byproducts like the globalin, the cruciferin, and the napin, both fractions we have to use. And in this case, the challenge is to reduce the taste, to improve the taste, as Gertjan said. And that is a challenge. And that was a challenge for the last couple of years, and still it is. So if we talk about good quality, we don't have to forget the pricing. And I'm absolutely confirm Søren regarding his concerns regarding the pricing. Otherwise, that product will never fly. It's more than comments than a question. Thanks.
So do you want to answer the comment, Gertjan?
For me, yeah, no, I fully agree with what Stefan was saying. Further improvement in yield, in protein level, also being able to get all the protein out of the material, that will help. And also there with breeding, you might be having some improvements and taste improvement. And indeed, as he says, you need niche projects, or at least you cannot make a commodity that is competitive. You really need to have specific application fields where you can use it right now. That might change in future if we are developing in the right direction.
I think that will be the end of our session today. It was, thank you, everybody. It's past 9 and we're supposed to go to 9. So it was a very interesting session. I learned a lot. Thank you for all the speakers. Thank you for all the people that asked the question. I think it's a complex issue. Protein canola is a complex issue. I think we will have to change a lot of things because the processing that DSM is using and the processing that Dave Disiak was talking about, they go from the seed and DSM are going from the cold press. And most of the canola actually right now is what I call conventional press, which is expeller press, solvent extraction, toasting of the meal. So if at the end of the day, like if I take Canada, we're crushing 9 million tons of canola, and we're crushing 9 million tons of canola conventionally. So there is also some research and some improvement that has to be done on the processing to have actually a meal that's going to be able to be used by a lot of people and a lot of processing, because leaving a lot of the oil in the meal, like when you do in cold press, is not very efficient. So I mean, it's done, it could be done, but you're losing a lot of money. And when the crushing margin are really low, you want to extract as much oil as possible from a crusher. So I think actually it's a very interesting, we have to look at the big picture. Today we look at the breeding and we'll look at the processing of the protein, and maybe there is a piece missing, which is actually the processing, the conventional processing of the seed, and what could be done actually to increase the value of the meal that's produced, so that meal can actually be processed in something else and can be processed to produce good quality protein. Because for the moment, because of the toasting, phenolics bind to the meal, and it's not really that easy to use. So I think that will be for the next meeting and the next session on protein and meal quality and quality of the canola. And I really, really want to thank everybody that's been participating, everybody that's been attending, because it's been very early for some of us. It was very late for other people, depending on where we are in the world. So it was morning, afternoon, night. And I think this is the great things about the virtual meeting. We can be anywhere in the world and taking it from home and taking it almost any time of the day. But I really want to thank everybody to be there and then participating and asking questions. It was a great meeting, and I really I hope everybody enjoyed it as much as I did. Thank you so much. And Wolfgang, if you want to close the session of Etienne, if you want to say a last word, that will be great. Thanks.
Yeah, thank you very much, Veronique. Thanks particularly you for organizing this very interesting session. We had a very good discussion, I think, and really the aims of the technical meeting really were achieved. We had very stimulating talks and discussions, and that is particularly due to your preparation, Veronique, and Sam Cooks. And we also thank Ivona Bartkowiak-Broda, who actually was the organizer of this technical meeting. And we really appreciate that we could have this online version, not optimal certainly, but was the best we could have at this time. Obviously, we will have another meeting tomorrow. That is actually the GCRSC General Assembly. That is only for members, of course, but those of you who are not members yet can become members, of course. So you are cordially invited to become a GCRSC member. It's rewarding, I would say. So I would recommend and invite you to do this. And with that, I would like to conclude this technical meeting, and thanks everyone for participating and contributing to the very interesting discussion, which certainly gives hope that we will make progress in the coming years, and will be interesting to see what has been achieved in 2 years' time when the International Rapeseed Congress is going to take part in Sydney, Australia. Thanks very much.
Thank you, Wolfgang. And yes, I'm really looking forward for the next face-to-face meeting and see all the people, not just on the screen, but see them and talk to them face-to-face. Etienne, you have the last comment?
Oh, I think Wolfgang closed very well this meeting. So thanks to all. This is my only word, I think, because this job is the fact of the effort, in fact, of all GCRC members, and I mean speakers from everywhere, in fact. So I think this today were really of good and great quality. Yeah. Thanks.
Yeah, I really appreciate the effort of all the speakers. Some of them has issue with presentation and loading, and Gertjan, I really appreciate you coming in when Sama could not do it, and almost at the last minute, and be so, doing such a good job and presenting everything and no preparation. So thank you so much for that. It was really, really appreciated, because processing was a big important part of this meeting and so new for us. So thank you again for that change, and thank you for that. And last but not the least, I would like to thank Michelle, because Michelle was in the background doing all the registration, all the monitoring and helping all of us with this technology that's good when it's working and so bad when it's not. So thank you for that. You're in the background, but as I say, you had a very long day, starting early and finishing late because all of us are different timetable. So thank you for that. It was greatly appreciated also. So with that, have a good afternoon, good evening, good day, I'm not sure. And I hope to see all of you at the meeting in Australia when we will be able to travel and meet again in a new life after COVID. Take care, guys. Nice to see you.